Bismillahirrahmanirrahim. Today we'll talk about the muscular tissue. Muscular tissues are responsible for moving the body and body organ. For example, when we walk, actually the muscles of the skeleton contract and relax to let us walk. And this happens when we want it. So it is a voluntary movement. And so the skeletal muscles are voluntary muscles. The second type of muscles is found in the heart. And the heart muscles are continuously moving and contracting to pump blood through all the body. We cannot control the movement of the heart muscles, and so the cardiac muscles are involuntary. That is, we cannot control them. The third type of muscles is found in the inner viscera, or the hollow organs within our bodies, and these have movement or they move the material within the hollow organs, and we cannot control this movement. So it is involuntary. Now, the cardiac muscles are discussed with the cardiovascular system. And today we are left with two types of muscles: the skeletal muscles and the visceral muscles. When we take a microscopic section and look under the microscope, we'll find that the skeletal muscles are formed of cells which are long cylindrical fibers, and they are striated with cross striations or cross lines on each fiber. So we call it striated skeletal or striated. While the visceral muscles in a section do not have these striations, so we call them smooth muscles. So we have visceral muscle, visceral or smooth, and skeletal or striated muscles. Let's take the first type of muscle. But before that, we'll talk about certain words which are the terminology which we will use with all the types of muscles. Myos in Greek means muscle. So myo, muscular. Sarc in Greek is or means flesh. So sarco means fleshy. Myosite, myo, muscle, site, cell. So this is the muscle cell, and we will call it the muscle fiber. Sarcolemma is the membrane of the muscle cell or the muscle fiber. Sarcoplasm is the cytoplasm of muscle cell or muscle fiber, while sarcoplasmic reticulum is the smooth endoplasmic reticulum found in both types of muscles, and they are very important in muscle contraction and muscle functioning. It is given the name sarcoplasmic reticulum. Now, let's take the first type of muscles, which is skeletal muscle, because it's found in the skeleton, and it is voluntary because its movement is under our will, and it is striated because it appears striated under the light microscope. If we take the muscle that you see here in between the two bones, moving the bone, we'll find that it has tendons at the end and the muscle in the middle. The muscle itself is covered by connective tissue covering called epimesium, which is connective tissue continuous with the tendon. As you know, the tendon is collagenous connective tissue, and so the epimesium is continuous with it. This epimesium allows a muscle to contract and move powerfully while maintaining its structural integrity. That's one thing. And as you will know later, it is very important in the contraction of the muscle. Now, in a cross-section of the muscle, we'll find that it is formed of group of fibers, which each of these groups is called the fascicle or nisira. These fascicles are also surrounded or covered by connective tissue covering, and this connective tissue covering is called perimesium. Peri means around, hawla. Perimesium, around the muscle. Each fascicle, if we take the fascicle, we'll find that each fascicle contains a large number of muscle fibers or muscle cells. The muscle fiber or muscle cell is a cylindrical long cell or long fiber, which is covered also by connective tissue covering called endomesium. In endomesium, perimesium, and epimesium, collagen is continuous with each other, so that any pulling of collagen in the endomesium by the muscle fiber will be transferred through perimesium to epimesium to the tendon to pull the tendon. Please remember that until the end of this section. Now, skeletal muscles are having a large number of or large supply of blood vessels, which are, as you know, found in the connective tissues of the epimesium, perimesium, and endomesium. It also is supplied by innervated by a motor axon, which innervates each muscle fiber, as we will know later. In microscopic structure, when we have a longitudinal section in the muscle, we'll find that it is forming of elongated cylindrical muscle fibers or muscle cells, but the cell here, it does not have one nucleus. Each cell has several nuclei, and they are not in the middle as we expect, but they are on the periphery at the sides of the muscle fiber. If we take a transverse section in the muscle fiber, in the muscle itself, we'll find that, as we said, it is covered by connective tissue covering outside there, which is the epimesium, thick connective tissue, which continues down there to surround the fascicles, the perimesium. From the perimesium, connective tissue is continuous with another connective tissue surrounding each fiber, which is called the endomesium. We said that the muscle fiber is cylindrical and it contains a large number of peripheral nuclei. We said also that the fiber has blood vessels. Large blood vessels are found in between the fascicles in the perimesium, and small capillaries can be found around the muscle fibers or the muscle cells themselves in the endomesium. There are two types of muscle fibers. As you will know in anatomy, they may be found in certain muscles, or they can be found in different muscles mixed together. One of them is the red muscle fiber. The second is the white muscle fiber. What are the differences between red muscle fibers and white muscle fibers? The red muscle fibers in an electron micrograph appear smaller diameter with a large number of mitochondria, while the white muscle fibers have larger diameter and a small amount of mitochondria. Under the light microscope, we'll find that the red muscle fibers are dark red due to the presence of myoglobin, the special protein found in the red muscle, giving them the red color. And the white muscle, the muscle fiber itself is larger and it is paler in color, having more glycogen. The white muscle fibers are easily fatigued. Why? This you will know in biochemistry and physiology. Now, when we stain these muscles histochemically for the activity of succinic dehydrogenase in mitochondria, which shows the presence of mitochondria, we'll find that the red muscle fibers are densely stained blue, as you see, and they have a large number of mitochondria, while the white muscle fibers have less activity and so they have less mitochondria. When we stain ATP histochemically, we'll find that the red or slow muscle fibers are stained very dark due to the presence of large amount of mitochondria of ATP produced by mitochondria, of course, while the white or fast muscles have paler stainability, showing that they have low amount of ATP. Now, The myosite itself, we said that it is a long, maybe 30 centimeters, a very long, the one cell which is represented by fiber is very long, and it is striated, as we said, and it has a large number of nuclei. Now, I hope you will understand why it is long, why it has large number of nuclei, and why is it striated, and what is the importance of this striation during the next section. First of all, why it is very long and having a large number of nuclei? Because during development of the fibers, we'll find that they are formed of single cells which have single nuclei called myoblasts. The myoblasts fuse together to form myotubes. The myotubes fuse together to form a long cylindrical muscle fiber with a large number of nuclei. But one of the myoblasts stays as a stem cell in order to divide in case when it is needed, in case of anything happens to the fiber, okay, or degeneration occurs in the fiber, or it can also fuse its nucleus into the fiber to produce more proteins of the fiber and increases the size of the fiber in case of training. Now, if we take the ultrastructure of the muscle fiber, we'll find that in the cytoplasm there is a large number of mitochondria in the cytoplasm. And in the cytoplasm also there is a sarcoplasmic reticulum, which is the smooth endoplasmic reticulum, and it is abundant and unusually large, as you see here. Okay, why? We'll know that it is very important in the functioning of the muscle, and so it is like that. The sarcolemma or the plasma membrane is continuous inside the cytoplasm in the form of tubular structures which form a network of tubes called T-tubules or transverse tubules. Transverse inside the cytoplasm, tubules. These tubules, together with the sarcoplasmic reticulum, form what is called the triad, and we will know the importance of this structure during talking about its function. Inside the cytoplasm or the sarcoplasm, there are special structures which are found only in the skeleton, in this type of muscle, or and the cardiac muscle also, as you know. These are called myofibrils. This is the unit of functioning of the muscle fiber. Let's take these myofibrils, which are about more than hundreds and thousands found within the muscle, will find that they are formed of the special proteins of the muscle, which are the actin and myosin, which is found only in muscles, and they are in the form of myofilaments. Myofilaments forming these myofibrils. So the muscle fiber contains a large number of myofibrils, and the myofibrils are formed of myofilaments, actin and myosin myofilaments. Now, if we take an electron micrograph, we'll find that the myofilament is formed of several sarcomeres alongside the myofilaments. So the units on the myofilament is the sarcomere. The sarcomere actually is the unit which is important for the process of contraction. So let's take the structure of the sarcomere under high power electron microscope. In high power electron micrograph, we'll find that the sarcomere is the area found in between two dark lines called Z lines. Around the Z lines, there is a light area which is called light band or I band. I means isotropic because special microscope was used, and when light of this microscope passes through this part, it is similar to the light of the microscope which passes through. In the middle, there is a dark area which is called the dark band or A band. A and isotropic. An means not similar. So it's anisotropic or not similar to the light of this microscope, and so it appears dark. In the middle of the dark band, there is an M line, and around the M line is a lighter area, not as dark as the rest, and this lighter area is called the H zone. Now, let's look how these parts are formed. We said that the sarcomere is formed of the specific protein of the muscle, which are actin and myosin protein. Actin is actually thin filaments. Thin filaments which are mainly found in the eye band. These are actin or thin filaments. In the dark band, we'll find that these thin filaments, which are red, are found together with thick filaments, which are blue, and these are called myosin filaments. So we have actin filaments, thin filaments, found mainly in the eye band, and actin alternating with myosin in the dark or A band. the actin filaments are connected to the Z line, while the myosin filaments are connected to the M line. Now, the overlapping between actin and filament in the dark band is discontinuous, is not found in the H zone, which contains only myosin filaments. That's why it appears lighter than the parts around it. Okay. Now, muscle innervation. We said that muscles are voluntary. Skeletal muscles are voluntary. Voluntary because they are innervated by motor nerve. The motor nerve runs in the connective tissue of the Epimesium, and it branches in the endomysium to several terminal twigs. Each of them innervates a muscle fiber. When it innervates a muscle fibers, it forms a special structure which is called the motor end plate. Let's take the motor end plate and see the relationship between motor end plate and the muscle fiber. We'll find that actually the motor end plate makes a synapse with the muscle fiber, so that the nerve ending, which have a terminal bottom containing the synaptic vesicles, which contain acetylcholine, gets in contact with the membrane of the muscle fiber or the sarcolemma, which forms in this part invaginations called the synaptic clefts. The membrane of synaptic clefts has special receptors for acetylcholine on it. So, and of course, as you know, the sarcolemma or the cell membrane of the muscle fiber is continuous with the T tubules, which reach the area where the sarcoplasmic reticulum is found and forms the triad. Now, let's see these structures in action. When a nerve impulse comes in the motor nerve, it activates the motor nerve to produce acetylcholine, and this acetylcholine activates the acetylcholine receptors on the membrane of the or on the sarcolemma or the membrane of the muscle, and this leads of depolarization of the membrane, and this depolarization and activation continues through the T tubules to reach the triad, where it activates the sarcoplasmic reticulum to produce calcium. This calcium activates the two actin and myosin to slide against each other, and this sliding leads to contraction of the muscle, because sliding makes the two Z lines nearer to each other, and so contraction takes place. Now, this process, let's take here how this occurs. We have the myosin, the upper part with the head, the myosin and the actin myofilament down there. The myosin myofilaments pulls the actin in order to slide over it and shortens the distance between the two Z lines, leading to contraction. Now, three molecules are very important in this process. Let's look at them. Number one, we'll find that calcium is very important, and we'll know why. In the thin filament, there is a protein called troponin. Troponin blocks the myosin binding site, and this myosin binding site is found on actin, so it covers this binding site, and so myosin cannot get in contact with actin to pull it. But when calcium is attached to troponin, as you see here, the troponin will get up and release the myosin binding site on actin, leaving for the head of the myosin to bind to it. The head of the myosin has this black structure, which is ATP, which can give energy to the head to make its movement. Now, when this happens, the head binds to actin, head of myosin binds to actin, and moves to pull it, and so the slide. Let's look at it. The two sarcomeres. A sarcomere which is fully relaxed, and a sarcomere which is fully contracted. First of all, look at the Z lines. We'll find that the Z line in contracted sarcomere are nearer to each other than the fully relaxed, and so the sarcomere is smaller in length. Okay? Now, look at the I band, the light band. It is large in fully relaxed and small in contracted. How about the A band or dark band? We'll find that it is the same, the dark band. Why? Because the dark band is formed of the myosin filaments. Okay? But you see that the dark band, the part which is having actin and myosin together, is dark, and it is large and fully contracted, so the actin and myosin are together over each other, overlapping each other. But in the relaxed, they are not. And so the H zone, which have only myosin, is large in the fully relaxed, while in the contracted it is almost absent. How is the contraction of sarcomeres transferred from sarcomeres to the tendon, leading to muscle contraction? If we look to the sarcomere and the relationship between sarcomere and the membrane, remember, we have sarcomeres, hundreds and thousands of sarcomeres on every myofibril, and hundreds and thousands of myofibrils within the sarcoplasm. And all are now, let's see the relationship between them and the membrane. Here we'll find that there is a special protein called dystrophin. This dystrophin is connected to the myofilaments, actin myofilaments, and from one side connected to the actin myofilaments, and from the other side connected to a transmembrane protein called dystroglycan. Dystroglycan and dystrophin, both of them form a structure called costamere. The dystroglycans are connected inside the fiber, in the cytoplasm, to the contracting filament, as we say, actin, and outside the cell is connected to the laminin of basal lamina through the cell membrane. The basal lamina is connected to the collagen of endomysium. Now, we'll see that this structure, which is costamere, can transfer the movement of the myofilaments inside the fiber to the collagen of the endomysium outside the fiber. Remember that the collagen of endomysium is continuous with the collagen of perimysium, continuous with the collagen of epimesium, continuous with the tendon. So the movement or the pulling on the collagen of endomysium will result in pulling on the tendon, and the muscle can contract, as you see. Okay. Now, here, as you see, the pulling of the collagen around this, the pulling of the collagen of the endomysium results in the pulling of collagen of perimysium, of epimesium, and pulling of the tendon, and so contraction of the whole muscle is affected by the transfer of movement from the inside of the fiber by costamere to the collagen outside the fibers. Okay, now let's take the second type of muscles, which are the smooth muscles or visceral muscles or involuntary muscles. The smooth muscles are found, as we said, in the hollow organs of the body, such as the artery and vein of the circulatory system, the respiratory system, the bronchioles and the bronchi, the urinary system, the ureter, for example, the female genital system in the uterus, and in the gastrointestinal tract in the esophagus and in the intestine. In the intestine, we see that the muscles of the intestine are arranged so that the outer muscles are arranged longitudinally and the inner are circular muscles. In between is the ganglion cells of the autonomic nervous system. So both muscles can move the intestine in what we call peristaltic movement. You will know it when you take the intestine, and this will be affected by both the longitudinal and circular muscles activated by the myoenteric plexus ganglion cells in between, which represent the autonomic nervous system. Now, if we look to the muscle fiber in circular muscles, we'll find that it appears spindle-shaped. Spindle-shaped means thick in the middle and tapered on both sides. The muscle cells or the myofibers are arranged so that the thick middle part is adjacent to the thin part, and so they form the layer of muscle. The smooth muscle fiber has single nucleus, which is cigar-shaped and in the middle. This appears when we take a cross-section in the longitudinal muscles. We'll find that if the section passes in the middle of the muscle fiber, it will have the nucleus, while if it passes in the tapered part, it will have no nucleus. If we take the structure of the smooth muscle fiber, we'll find that the smooth muscle fiber have a cytoskeleton of intermediate filaments connecting the different parts of the cytoplasm by what is called the dense body to all through the cytoplasm and to the sarcolemma or the plasma membrane by what's called the dense plaques. If we take the dense body, we'll find that it is connected to actin filaments. And the actin filament is also connected to the sarcolemma by dense plaques, and remember that both dense plaques and dense bodies are connected by intermediate filaments. And remember that the actin filament is important for sliding over the myosin filaments, moving the part it has, and so by moving these parts and pulling on the intermediate filaments, the whole cell can contract. Actually, there are myosin filaments which are surrounded by actin filaments for the process of filament sliding and contraction, as we see there. But here you don't have the same structures that show in the skeletal muscle. On the membrane, there are invaginations called the cavuli, surrounded by the sarcoplasmic reticulum. Although the sarcoplasmic reticulum is not voluminous and is not large as it is in the skeletal muscle, but still it is there, and both cavuli and sarcoplasmic reticulum are very important in the movement of calcium for the sliding of myofilaments and contraction. These cells have a special characteristic that they can regenerate. For example, here we'll find that autoradiography with radioactive thymidine, which is a precursor for DNA, appears in some cells, indicating that they are capable of division. The cells are actually connected together by the intercellular matrix, which is connective tissue, and connected to the dense plate. So any movement in one of the cells will be transferred to the rest of the cells, and so they can move all together. There are gap junctions here, which we will talk about them later. In section under the microscope, we can see that the connective tissue is found in between the cells and connecting them all together. Now, let's start the activity. The activity starts with a neural stimulation. The autonomic nerve has at its end enlargement called neuron varicosity, containing the neurotransmitter. When neurotransmitter is released, it will activate the membrane of the fibers, and this activation of these fibers will be transferred through the gap junction and spread through to all the other fibers to be activated all together. What happens when depolarization of the membranes of these fibers occur? When this occurs, we'll find that the action potential results in diffusion of calcium through the membrane channels into the sarcoplasm of the fiber, and also the smooth endoplasmic reticulum or sarcoplasmic reticulum will release calcium. Calcium will bind to special protein called calmodulin. Calcium with calmodulin activates an enzyme called light chain kinase, myosin light chain kinase enzyme, which transfers phosphate from ATP to the light chain of myosin. When this sliding occurs, of course, the fiber will start contraction. All the fibers leads to contraction of the muscle of the organ, and this leads to the movement of material within the organ. Thank you.